So we're going to start with talking a bit about brain donation. We're very pleased to have a couple of people. I was going to call you speakers, but I hope it's more of a discussion than a talk. So Zeinab is one of the academic clinical fellows who's been based here for the last 6 months and is off doing clinical training, but will be coming back to us in a couple of years to do her PhD. And she's going to talk a little bit about the science, the reason why those of you who have been approached about brain donation have been. And then Susie Barker, who's our research governance manager here, will talk a little bit more about the practicalities. As I say, there are lots of questions that people often have, and this is a good opportunity to share any concerns or experiences that you've had and to find out a bit more about what's involved. So without further ado, Zeinab, if you're happy to come and introduce yourself and tell others about the science that you're doing, that'd be great.
Hello, nice to meet you all. I've been to one PCA support group before, so it's nice to be back. Right, so I'm one of the clinicians that does some research here at the Dementia Research Centre. So I'm just going to talk to you about the reasons why we've started conversations about brain donations, why as a clinician who's looking to do some research in the pathology of dementia, and why I've become involved in this area really, and just so I can share my learning with you. So this is really quite straightforward. The brain is one of those parts of the body that is really inaccessible, hidden away nicely to be safe in the skull, so a very complex part of the body. And really in lots of other diseases, as doctors, we can just put a needle in, get a biopsy if it's the liver, the kidney, whatever we want to have a look at, the lungs. Just do a biopsy, we get the results back, we can look at the biopsy under the microscope, and we can generally, with a very high degree of confidence, give a diagnosis. But the problem with the brain is that, as I said, it's difficult to access, and there's some quite big problems about doing biopsies on patients who are still alive. You can get risk of bleeding, risk of infection, and often it's really felt that it's not the appropriate thing to do. And we only really do brain biopsies in very rare circumstances when we think maybe that there is a very acute onset problem that can be reversed, maybe with antibiotics or with other drugs. So we tend not to do brain biopsies in patients who've been admitted with a longer chronic memory problem. So we really rely on diagnosis after death. So why do we need to study the brain? Especially in dementia. So we need to really be looking at the causes of dementia and to develop treatments. And doing a post-mortem and looking at the brain under the microscope at the moment helps us with understanding the molecular mechanisms of why people develop the dementias. And also we can do larger scale studies to look at really on a population level as to see why some people go on to develop dementia, some people don't, and why different kinds of people develop different types of dementia. And again, what we can do is study people during life, look at the brains post-mortem, and correlate the two findings and come to some conclusions from that. And really, in terms of what has brain pathology done for us until now, I'm sure you're all very widely read attendants here, often very well informed from my previous understanding, and I'm sure you know about amyloid beta hypothesis of the cause of dementia. And really these wouldn't have been found was it not for us being able to get to the brains and really study them post-death. So these are the references for the big studies that were done on amyloid peptide, and also tau, which is one of the major components of the neurofibrillary tangles. And again, that was discovered in 1988. And again, you really wouldn't have been able to see that just on a biopsy. So that's quite important. And just a couple of pictures as well. So on the right here is the tau positive neurofibrillary tangle looked at under a microscope, and over here is an amyloid plaque. And what we do is, once we've got the slides of the brain, we've got special stains that stain the slides, and then you can see, look, there's the fibrillary tangle, there's the amyloid plaque. Clearly this patient has an Alzheimer's disease pathology as a cause of their dementia. So it's really very useful and very widely used across the world. Really, why else would you want to do a post-mortem analysis of the brain? At the moment, the literature really says that the accuracy of the diagnosis of the different subtypes of dementia, depending on the centre, I'm sure at UCL we've got much higher accuracy rates than this 87%, But if someone comes to you with memory problems and they're diagnosed, we think we're accurate in life. About 70 to 87% of the time, we can say, actually, I think this is a Alzheimer's pathology, this is a frontotemporal dementia, or any kind of other illness. So, sometimes, obviously, as you can see, sort of up to about 30% of the time, once you do get access to the brain, once the person's passed away, you find that maybe the pathology is actually different. But again, at UCL, we like to think that we're accurate most of the time, hopefully. And really, an accurate diagnosis can have implications for family members in terms of genetic findings, and if that, if people want to know about that, then there can be a discussion once we know the results. So, really trying to conclude, is it still important to collect human tissue? You might think that, we're sort of science is advanced, why are we still having to look at the brains? And really, we do have animal models that can replicate the disease, but they're not really that representative, and they're not perfect. After all, it's just a mouse, and it's still different to the wonderful human brain. So we do still rely on having a look under the microscope at actual human brains. And really, in terms of new technologies that are coming on that we find that will be useful in applying to the neuropathology is, so I've just put a couple of things here. So RNA expression profiling is, we've normally so far been talking about DNA in Alzheimer's disease and in the dementias, but now we're starting to find that there are these, once the DNA is coded into the RNA and the mRNA, that there were these areas that we thought previously were just actually not used for anything, but there's some quite recent interest now looking at these supposedly non-coding areas of the DNA that actually do have implications on the way that your protein is made. And so there's some work going on there, and what you can do is you can actually measure the levels of these RNAs in the brain and see if that has an implication on the kind of disease that patients have. Proteomics is looking at the levels of certain proteins in the brain, and that helps us in developing biomarkers for trying to diagnose dementia in life. So, for example, if you measure certain proteins in the spinal fluids that we do often here, that's a biomarker, and we're all constantly developing new biomarker techniques, and proteomics helps with that. We need the brains to try and have a look at those proteins in the brain. And also, again, more in-depth DNA studies as well, which we can do under the microscope once we've got access. And also, obviously, what you want is we're constantly trying to develop new treatments for dementia, and once you've got a hypothesis, a new possible drug that you think might work, you can't just go and obviously try it on a live patient. So you can go to some brains that you think have been affected with Alzheimer's, you can try it on that and see if it makes a difference or not. And then that's called target validation, and it helps with trying out your new drugs and coming up with new treatments. And so really it is an essentially vital tool in trying to understand the pathology of dementia, understand the pathology of Alzheimer's, and really trying to come up with new treatments. And I'll stop there, and it was at this point, how did I come into this? So I'm a clinician who's planning a PhD in this department, and I'm always been really interested in pathology. And so when I started here, I had a chat with Seb about possibly doing some pathology work with regards to patients with PCA, because it's I'd actually never heard of PCA until I came to this department. It's you know a lot more than I do. And so wow, it's quite fascinating. It's such a different, unusual presentation of memory problems, and often doesn't start with memory problems. And so we started looking at the database of the brain bank to see, well, actually, how many PCA brains do we have to work with? And we have, at the last count, 7 PCA brains. And also at the last count, the brain bank has about 70 cases of patients with different types of dementia. So with those 7 brains, we can do some preliminary work. And just to explain some of the work that we were thinking of doing. So, for example, in some of the different types of Alzheimer's, the ones that present with a language variant, where people start having problems with language, you often find, because for the majority of people, the language area is on the left side of the brain, what you find is once you go into the brain and you compare the right side to the left side, that actually there's more tangles and plaques on the left versus the right. Now, Seb here is a clever man. He knows, well, actually, why don't we go and look at the right side in PCA, which is where the visuospatial pathways and areas are, and see whether there's a greater tangle pathology and a greater disease burden on the right versus the left. So that could be one of the studies that we could do with those 7 brains to start with. It hasn't been done until now. And also, again, that's important because when you have a look at the brain on MRI scans, you can see that the pathology, the right area of the brain is more affected compared to the left, which is why it does make sense to then go on and do these studies. And other studies that we've been planning on doing possibly is when you look at people who don't have a diagnosis of Alzheimer's disease, no memory problems at all, once you look at their brains on MRI scans and on certain specialized scans, which we call DTI, which looks at the pathways of the brain, so it really visualizes the tracks. They've shown in patients with no memory problems at all in this at UCL actually that the size of the certain tracts going from the back of the brain to the front of the brain actually do have a bearing to the visuospatial processing skills of that person, whether they have Alzheimer's or this within in healthy volunteers. So we wanted to try and extrapolate that to see, well, actually, why don't we look at that part of the brain under a microscope and see whether that bears any reality in patients with PCA, that maybe this sort of asymmetry of these pathways, maybe that's the pathway along which PCA kind of propagates, if that makes sense. So if the right is more affected than the left, and if the pathology sort of starts at the back of the brain and comes forward, if that makes sense, along the right side of the brain. And also, finally, another thing that we were interested in, again because of the striking visuospatial processing problems in PCA, there's been studies shown using it's called hypometabolism on these scans, which we call PET scanning. They've looked at different parts of the brain, and when they look at the parts that control eye movement at the front of the brain, funnily enough, they've shown that those parts of the brain actually have reduced metabolism, as well as the back part of the brain, which you would expect in PCA because of the areas of visual processing. So what we wanted to do is, again, have a look under the microscope at the back of the brain and at the front of the brain, which controls eye movement, again, to see whether there is maybe at those parts of the, and which this has never been done before, to see whether those parts of the brain at the front which control your eye movement are also affected in PCA, and how we can link that to the back of the brain. I hope that makes sense. I didn't have any slides for anything, I just wanted to have a discussion about it. So there's lots of things we can do that have never been done in patients with PCA, and this is obviously a centre in the country that does lots of work with PCA pathology, and we have obviously a great group and network of patients. So that's how I came into it, and really, so I just think as a clinician, having access to what's really such a precious resource is quite incredible, and I'm really sort of blessed that we have 7 brains to work with, and we can do quite a lot with that. But that's, the reason I've come here to talk to you is to really tell you about my experience, tell you about what I've been doing, and just to start a dialogue and about brain donation. And if anyone is interested, Susie has some of the more practical aspects of how we might go about it. But we're always very grateful. We know it's a very big decision to go on to donate brains, and but I just need you to know and want you to know that it's also extremely, we know it's precious, and we know that it's not, if you've made the decision to go ahead, it will not be wasted, and it's very helpful in coming up with treatments and understanding the disease. Okay. All right.